All right, hello. I'm going to run through the Chapter 12 review that we would have done today in class if we'd had a normal class day. And the PowerPoint that I'm using for the review is, of course, going to be down here in the Unit 3 exam review and practice. So I'm going to click on that. I have had a couple of people ask me about discussion questions on this exam. There is no discussion question. What is going to substitute for a discussion is the genetics problem from Chapter 8. And again, for that problem, I'm going to give you a messenger RNA sequence. So a bunch of C's and G's and A's and U's. And all you have to do is find the AUG, start there, that codes for methionine, look up, read the next three, the next three, the next three, the next three, look up the amino acids on the table, write it out as a chain, just like we did with those activities in class. And then when you hit a stop codon, you're done. That's all there is to it. That's going to be, I haven't made out the test yet, but that'll probably be 7, 8, 9 points, something like that on the exam. And here are the Chapter 8 and 5 review questions that we went over. Here's Chapter 12, so this should be available to you. This review is not super long. Chapter 12 is kind of the easy part of the immune system. The harder part comes in with Chapter 13, which we started covering today in class for Unit 4. So that's the tougher chapter. Chapter 12 again focuses in on start our slideshow here. A general overview of the immune system, and also the assistance it receives from the cardiovascular system, especially the blood vessels, and then also the lymphatic system. So we did talk about the roles that those play with immunity. So kind of an overview. Again, the goal of the immune system is to eliminate things that are non-self, foreign to the body. And in Chapter 12, we mainly focused on non-specific immunity, which includes your first and second lines of defense. So these include things like barriers, cells, processes that you're born with, so you don't have to be exposed for them to kick in. They react right away. They're available. And they defend us against microorganisms and other things that are foreign non-specifically. So just they have ways of finding bacteria in general, fungi in general, that type of thing. When we get into Chapter 13 and we're looking at specific immunity, that's where we generate these very powerful lymphocytes and antibody weapons that attach to very, very specific molecules on particular kinds of microorganisms. And that's more powerful and more complex, and part of Unit 4 rather than this unit. Okay, so again, with you just keep in mind, we know the blood is involved with immunity because your white blood cells, the different kinds, which we've talked about the basic functions in Chapter 12, that's how they travel throughout the body. They are able, remember, to get out of the blood and go into tissues, kind of like they're showing you here on this particular diagram, where they can help us fight infections, repair damage, trigger inflammation, those types of things. And then also we have this system of lymphatic vessels and lymph nodes and the spleen that also play a role, because the lymphatic vessels take up excess tissue fluid. Fluids come out of the blood, they go into our tissues, they become our interstitial tissue fluids. And then as those fluids build up to high levels and higher pressures, the excess drains into the lymphatic vessels. And so if you have an infection here, you've got some microorganisms in here, they're also going to drain into the lymphatic vessels. Those lymphatic vessels carry those fluids slowly to lymph nodes. And as we talked about in Chapter 12, lymph nodes are locations where we have lymphocytes and macrophages stationed to intercept microorganisms that may be draining off of an infected tissue somewhere in the body. So by doing that, then the lymphatic system is partnering with your immune system and playing a really important role in immunity. Eventually, so if this is a lymph node that's been cut open here, the fluid comes in, passes through here, and it exits through what we call an efferent lymphatic vessel or lymphatic duct. And eventually all of those fluids drain back into the left or the right subclavian vein. So the fluids actually get returned to the blood after they have been filtered and checked by the immune system. So again, most of chapter 12 deals with the first line of defense and the second line of defense. First line of defense, and you do want to outline what's involved with which particular branches of the immune system, which particular lines. The first lines are your skin and your mucous membrane linings, and there are chemicals and things like pH differences and mucus and things like that that help prevent microorganisms from living on those barriers or getting across. And when things do get across the skin and the mucous membrane linings, then they face the various white blood cell weapons and also some various processes like inflammation and phagocytosis. They're going to help clear infections out. And then we also talked about the complement system, which is a protein-based system that reacts non-specifically within the second line against invading microorganisms. So you do want to kind of organize those different things. Let's take a look. I'm going to pause here for a second because I need to flip my blinds on my window here in my office. All right, so make good use of the study guide / learning objectives for this chapter as you're going through to kind of help you focus on the important things. So some example questions here. Number 1, this antibacterial enzyme attacks bacterial cell walls and is secreted in your perspiration, your tears, your nasal mucus, and your saliva. So those are all fluids that you have on your skin or your mucous membrane linings. So this enzyme is part of your first lines of defense because it is a weapon in one of those locations. And so if you guys remember from class, we talked about how we do have an enzyme that digests peptidoglycan cell walls. And hopefully you remember that that is lysozyme. Yeah, not lysosome, but lysozyme. That's specifically what it does, so I would file that away in memory. We also did talk a little bit about dermacidins. Those are hole-punching proteins present in the skin that attach to the membranes of microorganisms and poke holes in their membranes. So that's another type of first line of defense weapon that we have sitting in the skin. Okay, this was a frequently missed question from the homework, and again, it looks like a lot of this, there were some questions that were missed that involved categorizing things that are features of the defenses. So let's see, this is one of those check all that apply type questions. Innate, non-specific physical barriers of host defenses. And again, with these, go down the list. So include the skin. Yes, we want to pick that one. Include the mucous membranes. Yes, those are part of our innate, non-specific barriers. Those are first line. Now this question doesn't specify only first line. It just says innate, non-specific. So that's first and second line fall into that category. Do they all contain keratin, which makes the barriers highly impervious? So impervious means that microorganisms can't get across too easily. And no, you would not pick that one because your mucous membranes are not covered with keratin. That's the skin only, so we don't pick that one. Innate, non-specific physical barriers of host defenses. Well, I'm sorry, this is only referring to the first line. Are they part of the second line of host defenses? No, because we're talking about barriers. Are they all covered with mucus? That adds another layer of protection. And so that one is kind of like this one. You know, the problem where there is all. So your mucous membrane linings inside the body do have mucus that helps trap microorganisms, but the skin does not. So you don't have mucus on your skin, I hope. So actually you would only pick those first two. First line defenses. Which of the following are correct descriptions of the first line of defense? All right, so again, we go down our list. Involves barriers at portals of entry. Yes, definitely. Includes chemicals such as lysozyme and lactic acid. Yes, we would pick that. Lactic acid is present in some of our secretions like perspiration. It helps lower the pH. Remember, we did talk about how you have a slightly lower pH on the skin And that helps prevent the growth of a lot of microorganisms. Involves phagocytic cells, neutrophils, and macrophages. We're not going to pick that one because that's second line. So those don't really get involved until you get past the skin or a mucous membrane lining. Then you face those kinds of cells. Involves recognition of foreign antigens by their pathogen-associated, or foreign agents, by their pathogen-associated molecular patterns. That's not part of your first line, that's second line. So we talked about those PAMPs and how those are molecules that are present on, say, like bacteria in general. So they're things like peptidoglycan. Lots of bacteria have that. And your white blood cells have receptors on their surface that are called PRRs, pattern recognition receptors, that bind those types of things and help them react or do phagocytosis, take up things by phagocytosis. But those are part of your second line, not first line. Examples include the ciliated epithelium of the respiratory passages, excretion of urine, and production of tears. Yes, those were all features of your first lines of defense that help keep things from getting across the skin and the mucous membrane linings in the first place. Now this series of questions was actually associated with a video, a little animation clip that was part of your homework, but we will, it looked like this was one of the lower scoring series of questions, so we'll run through this real quick. Which of the following are second line defenses? Check all that apply. So we're not going to pick lysozyme. We just said that's first line. Antibodies, we're not going to pick that. That's third line. So we're going to talk about antibodies in chapter 13. Interferon, yes, we will pick that one. And we mainly talked about, there's more than one kind of interferon. We talked about the type where if a cell is infected with a virus, or it could be another type of microorganism as well, inside the cell will send interferon signals to neighboring cells so that they will start to make antiviral proteins. And those antiviral proteins inside the cell will block the virus if it attempts to infect the neighboring cell. So those are part of your second line. Inflammation is a process of the second line. Fever is a process of the second line. Complement is also part of your second line. That is a set of proteins in your blood all the time that set off a train wreck. They set off a chain reaction when an infection with a microorganism is detected. That was called complement. So we talked a little bit about that last week. Examples of PAMPs, pathogen-associated molecular patterns, include, so how do you think about this? What are molecules that we have talked about in this course that are present on lots of microorganisms, but they're not our own molecules? PAMPs are going to be things that are foreign to the body. So interferons, no, we're not going to pick that because interferons are chemical signaling molecules of the immune system. Peptidoglycan, yes, cell walls of bacteria. That's not a human molecule, so that is going to be a PAMP that your immune system can home in on. Toll receptors, no, that's another name for pattern recognition receptor. That's kind of an older term. Complement, no, complement is a set of protein weapons that are part of our immunity, second lines of defense. PAMPs are molecules on the invading microorganisms. They're not our molecules. Lipopolysaccharide, absolutely. Where do you find that? Outer membrane of gram-negative bacteria. All right, number 3, fever and inflammation are normal body responses to injury or infection, and it may be best to withhold treatment in some cases. Absolutely, yeah, we talked about that in class. Yeah, even though most of the time we want to take medication for even a low fever so that we feel better. If you can avoid it, it's probably helpful if you have a low fever to just let the fever run its course. Fever does have some benefits to immunity. The membrane attack complex is part of the blank system. And that is part of your complement system. So again, complement is a set of proteins And when they detect a microorganism, there is basically a chain reaction. One step leads to the next, and membrane attack complexes, which are basically pores, get stuck in the membranes of microorganisms and can help kill them, clear them out of the body. What element is essential for microbes and can restrict the growth of pathogens when bound by antimicrobial proteins? So you may remember we talked a little bit about iron. Microorganisms need iron just like we do. And one of the things that your second line of defense does to help prevent the growth of microorganisms is soak up and hide iron from them. Here's another series. This was another one of those where you had to watch a video and then answer questions about it. And interferon only works against viral infections. That's actually false. Now, we mainly talk about it as being involved in viral infections. So I kind of don't like that question because we talked about it specifically as an antiviral. However, it can also help work against other infections where you have microorganisms inside your cells. There also are other kinds of interferons that play different roles in immunity. So I kind of didn't like that particular question from your homework. What is the major mechanism of action for interferon? So if you go back and look at the interferon story, interferons are released by cells in your body that have a virus inside them. They release the interferons, they secrete them, they go out, they attach to receptors on neighboring cells, kind of like a hormone attaching to a hormone receptor, and then that triggers the neighboring cell to activate genes for antimicrobial proteins, which they really should have said proteins instead of compounds there in that particular question. That would have been better. Those antiviral proteins, what they do is they prevent the cell from doing translation. So if the virus comes in, it can't make its proteins because translation has been temporarily shut down. So that's a way of protecting neighboring cells from a viral infection. The cell that produces interferon is protected from the infectious agent. That one is false. So an infected cell with interferon is sending a signal to neighboring cells. It's the neighboring cells that wind up getting protected. I'm going to scarf down some coffee here. All right, defense proteins in the blood. The blank system of blood proteins acts to lyse foreign cells and viruses. That was complement. So those membrane attack complexes, they can actually attach to the surfaces of enveloped viruses because they have a little membrane covering, remember from chapter 5. So you can get those holes punched in the membranes of enveloped viruses as well. Complement can do that along with poking holes, say, in the membranes of bacteria. The 3 complement cascades are activated by different mechanisms. That's true. We talked about there were 3 different things that can trigger complement activation. One was antibodies, which are a third line of defense weapon attaching to the microorganism. That can set off the complement chain reaction. And then there were also a couple of other pathways, the alternative pathway and the lectin pathway, where we have proteins in your blood and body fluids that can attach to certain microbial surface molecules. Lectins attach to sugar patterns. So certain combinations of sugars that are present on microorganisms, and those also will set off the complement set of reactions. Once complement gets activated, if you go back and look at the story, the same end results of the membrane attack complex, enhanced phagocytic capabilities, enhancing phagocytosis through opsonization, coating, and amplification of inflammation, making inflammation ramp up a little higher. So that is true. If you go back and look at the benefits of complement, once it gets activated, those 3 things are benefits. All right, here's another one. These proteins present at barriers attach to microbial cell membranes and create pores in them. If you go back and look at how things work, those were defensins. So those are hole-punching proteins that are located at our barriers and help poke holes in their membranes. The process by which white blood cells squeeze through blood vessel walls and enter tissues is part of an inflammatory response. So ordinarily your white blood cells stay in the blood. That's how they travel around. If you have inflammation getting triggered, you have an infection, an injury, then remember your local capillaries, microscopic blood vessels dilate, white blood cells can squeeze through, and that process has a name. Diapedesis was the name. Chemotaxis is where once the white blood cells come out, they travel, your phagocytes especially, are traveling kind of a chemical trail of breadcrumbs toward, a bunch of bacteria that might be present in an infected tissue. These white blood cells circulate in the blood as monocytes and then mature into wandering cells upon entering tissues. So if you go back and look at your white blood cell functions, those are going to be your macrophages. Monocytes in the blood become wandering macrophages. Once they do diapedesis and get out into an infected tissue, they transform into macrophages and they wander around and do phagocytosis. What is the function of a phagolysosome? So go back and look at the steps of phagocytosis. And what's going on with a phagolysosome? Phagosome is a little membrane-covered compartment that has the little microorganisms in it that got ingested. That's a phagosome. Lysosome, remember, is a little membrane-covered compartment full of toxic chemicals and digestive enzymes. When those two merge together inside the phagocyte, you now have a phagolysosome. So what's going on in there? So hopefully you're picking D here. You got the merging of the two. Now the poor little microorganisms that got ingested are going to be digested and killed and destroyed inside the phagolysosome. These cells have long appendages that capture microbes and are important for both innate and acquired immunity. These are the spidery-looking cells. Those are your dendritic cells. They're good phagocytes. They're coming back in chapter 13. They're also called antigen-presenting cells, and they help activate our T cells. But that's a chapter 13 story. Number 8, you notice redness developing around a mosquito bite. What is causing the redness? We talked about this in class. When you see redness, like on a mucous membrane in the back of your throat, for example, or on the skin if you have a lighter skin tone especially, that is caused by vasodilation. Inflammation causes local blood vessels to dilate. That's where that increased blood flow makes the mucous membrane lining or the skin take on a reddish appearance. It's not this one, fever. A lot of students wind up picking that. That's the most common incorrect answer that people choose. It's not fever. You don't have, when you feel warmth around an inflamed location on the skin, that's not fever. That's just increased warmth due to the increased blood flow. A fever occurs across your whole body at one time. That's increased body temperature that's been deliberately triggered. Okay, bacteria that drain into lymphatic vessels from tissues would likely face lymphocytes upon reaching. So remember, if you have an infection in a tissue, excess fluids are going into your lymphatic vessels, and if bacteria are there, they're going to drain into those lymphatic vessels as well. And where do they get picked up by lymphocytes? That's going to be in the lymph nodes. Remember, the lymph nodes are going to filter that lymph. And then once the filtered lymph leaves the lymph node, it's going to eventually be dumped back into the bloodstream in the subclavian vein. So it winds up getting returned back to the blood. This body region is protected by fatty acids, and acidic pH, lactic acid, and a tough cell barrier with its own normal biota? That's going to be the skin. I'm guessing, I'm kind of wondering, probably an alternative answer that people like to pick with this one could be the digestive tract. And that's probably because of the acidic pH, which we associate with the stomach. But just remember, the inner lining of the stomach, yes, does have an acidic pH, but the vast majority of your digestive tract has a normal pH. It's just the stomach that has that. So these are all characteristics of the skin, is your correct answer there. Like those fatty acids, for example, are coming from your skin oil or your sebum. Which of the following is not a type of phagocyte? So we talked about, of course, neutrophils and macrophages are your main types of phagocytes. But then we also talked about there are some specialized macrophages that reside in particular tissues. And one of those was the Kupffer cells. Those are in the liver. They're always stationed in your liver. And because the liver, blood draining off of your digestive organs and the spleen goes first to the liver. And so the liver is a great place to intercept any microorganisms that might be in that blood draining off of those organs. And monocytes are phagocytic. They're in the blood. Monocytes in the blood become wandering macrophages in your tissues. So all four of those are phagocytes. The lymphocyte here is not. So the correct answer there would be lymphocyte. Let's see, just a little review about a really appetizing topic, pus. Maria uses a microscope to examine a sample of pus from a pimple. Well, that doesn't exactly look like a pimple, does it? So this is like an ingrown nail. What should she expect to find in the sample? So remember, if you go back and look at why pus forms, if you in a pimple, you have a small amount of pus. And if you guys remember, with pus, we said pus was a sign almost always of a bacterial infection. And then think about, okay, well, what white blood cell type did we talk about being bacterial slayers, strongly attracted to bacteria, very abundant in the blood? Those are your neutrophils. So neutrophils swarm in. They're attacking the bacteria. They die. Neutrophils don't live for very long. So then more neutrophils come in. And all that stuff builds up with the excess fluid, the dead bacteria, and damaged tissues and so forth. And that's what creates the pleasant stuff that we call pus. Again, a pus is a sign of a bacterial infection. All right, something we talked about a little bit earlier in the semester. I don't think we actually talked about it during chapter 12, but diabetics and susceptibility to infection. Just as a reminder, this is just helping us kind of think through some things that we've studied in chapter 12. Diabetics experience damage to small blood vessels over time, which can impair blood circulation in extremities such as the feet. How might this affect their vulnerability to infection? So most of you guys are planning on going into nursing, and so this is something that's really common in diabetic patients, and we have a lot of diabetic patients in the United States, especially patients with type 2 diabetes, which is caused from insulin resistance. Your cells are ignoring insulin signals that tell them to take up glucose out of the bloodstream. So we wind up with too much glucose in the blood, which can be toxic and can wind up doing things like causing your blood pressure to be too high, little tiny blood vessels in places, especially in the extremities like the feet, become damaged. So what's going on? So these patients tend to have a really hard time healing wounds in places like their feet. They have problems getting over infections in places like that where they have really poor circulation. So why do we think that might be? So hopefully you are thinking about what goes on with those second lines of defense. So if you had a little tiny cut here and some bacteria got in this area on your foot, if you're healthy, remember you're going to have vasodilation, little local blood vessels will dilate, out comes the white blood cell army, Good stuff like complement comes out, helps attack that infection just with the second line. And hopefully all those little microorganisms that might be in there are going to get killed right away, and you heal this wound pretty quickly. You know, inflammation also involves tissue repair and wound healing. If you're diabetic and a lot of the local blood vessels are dead in this area, it's very difficult to get that white blood cell army into that area. It's very difficult to get nice things like complement to come in to help kill microorganisms that might be in a little teeny tiny wound. Well, then they can start to overgrow, and you can wind up with a really nasty wound that's really difficult to heal in those locations. So it's all about that poor circulation and not being able to fight off the infection quickly and heal the wound. I know the answer here is missing fewer antibodies, and we'll talk more about that in Unit 4. Antibodies are weapons of the third line of defense that also need to get out of the blood and come into an area of infection to help clear out those naughty little microorganisms that might be causing an infection. All right, so those were some example questions. Again, I think what we covered in Chapter 12 is not all that heavy duty. A lot of it's just, some of it's a rehash from AMP2 if you've had AMP2. So just kind of follow along. Make sure you have things, follow along with the study guide and the learning objectives and the PowerPoints and your notes from class. Make sure you have things categorized, like barriers, first line versus cells and processes and protein weapons and so forth in the second line. And you should be good with Chapter 12. The harder chapter for Unit 3, of course, is Chapter 8. And kind of the same thing. You just want to go through and use those learning objectives / study guide to kind of guide you through what you're reviewing for the test. So good luck. If you have any questions about anything from any of the chapters, Chapter 8, 5, or 12 on the Unit 3 exam, just get in touch with me so I can help you. Good luck. See you Thursday.